A study led by Duke Health has found that the antibiotic ceftobiprole, known for its effectiveness in treating bacterial pneumonia, is also promising in combating methicillin-resistant staph infections. The study, named Eradicate, involved 390 patients with complicated staph infections and found that both ceftobiprole and the antibiotic daptomycin performed similarly in terms of overall treatment success. This discovery offers a potential new line of defense against a common and often deadly bacterial infection, Staphylococcus aureus. If approved by the FDA, ceftobiprole could address a critical need for treating such infections, as there hasn't been a new antibiotic approved for this purpose in over 15 years. A study led by Duke Health researchers has shown that surgically joining the circulatory systems of young and old mice through a process called heterochronic parabiosis can slow down the aging process at the cellular level and extend the lifespan of the older mouse by up to 10%. The researchers found that the longer the animals shared circulation, the longer the anti-aging benefits persisted after they were separated. This suggests that young animals have components and chemicals in their blood that contribute to vitality and could potentially be isolated as therapies to rejuvenate the body and extend the lives of older individuals. The study offers valuable insights into the mechanisms of aging and the potential for anti-aging interventions. A study conducted on twins by Duke Health researchers has found that even a single traumatic brain injury, TBI, at any age is associated with worse cognitive function in later life, independent of genetic and environmental factors. The research indicates that TBIs, including concussions, can lead to a faster rate of cognitive decline in later years, particularly if the injury occurs after the age of 24 or results in a loss of consciousness. The findings emphasize the importance of treating TBIs appropriately and the need to minimize or prevent these injuries to potentially delay or prevent the onset of dementia. Researchers from Duke Health have developed a blood test capable of detecting Parkinson's disease, which could lead to earlier diagnoses and the initiation of treatments sooner. The test focuses on DNA damage in mitochondria, which has been associated with an increased risk of Parkinson's disease. Using polymerase chain reaction (PCR) technology, the assay identified higher levels of mitochondrial DNA damage in blood cells collected from patients with Parkinson's disease compared to those without the condition. Additionally, the test identified the damaged DNA in blood samples from individuals with the genetic mutation LRRK2 associated with a higher risk of the disease. The researchers hope this assay can help diagnose Parkinson's disease earlier and identify potential therapies to halt mitochondrial DNA damage. Researchers at Duke Health have developed an improved approach to CRISPR gene editing that enhances its functionality by identifying diverse CRISPR RNA variants that can specifically target challenging areas of DNA for editing. The new approach allows for more extensive genome editing, enabling the repair of mutations associated with a wider range of diseases. Unlike traditional CRISPR approaches, this method can salvage dysfunctional guide RNAs, making CRISPR technology more adaptable and efficient in its application. The researchers believe that this advancement will lead to safer and more effective CRISPR-based therapies for various diseases. Researchers at Duke Health have identified a fatty molecule in breast milk that could potentially reduce cerebral palsy in premature infants. In experiments with neonatal mice, this molecule was found to trigger a process in which brain stem cells produce cells that create new white matter, effectively reversing brain injuries. Cerebral palsy often results from infections that damage the brain's white matter, and there is currently no treatment for it. While further clinical trials are needed, the fact that the molecule is found in breast milk is promising, as it could be administered intravenously to premature infants with gastrointestinal issues who cannot safely consume milk or medication orally. This discovery holds potential for revolutionizing the treatment of preterm babies at risk of cerebral palsy.
If you enjoyed this video, please give it a thumbs up and subscribe to my channel for more great content.